clinical diagnostic. It tells you if the level of the hormone is normal, is high, or low. But the concentration itself is a correlate of the rate of synthesis, the rate of release, and the rate of degradation. So it's actually a dynamic value reflecting the interactions of these three processes. The concentration in blood for a hormone also may reflect whether it's the bound components or the free components. This is because most hormones, as I said, are actually carried in blood, bound to binding proteins. It's only the free form which is actually biologically active. When a hormone is bound to a binding protein, it's biologically inactive. So why do hormones bind to binding proteins? Well, for a number of reasons. If they're a steroid, they're insoluble in blood because it's an aqueous media. So steroid hormones, when they bind to proteins in blood, the concentration that can be carried in blood is increased. So the solubility is increased in blood. By binding to a protein, hormones are protected from degradation. They're also protected from clearance by clearance tissues like the liver and the kidney. Most hormones are very small molecules, and most will be removed after a single passage through the liver or the kidneys. So by having increased size attached to a binding protein, they actually survive for a longer period of time. So the roles of binding proteins are shown here. These letterboxes, by the way, should change in these are actually bullets which have changed from one version of PowerPoint to another. Should be bullets in your slides, I hope. Anyhow, these are the roles of binding proteins. First of all, it increases the solubility and the concentration of a hormone, providing a reservoir near the target site. Increasing the size of the hormone protects them from clearance and degradation, as well as degradation by plasma enzymes. Binding to a protein also inactivates the free hormone, thereby providing a buffer against large and sudden changes in the hormone concentration. Because hormones are released episodically, you get sudden increases in hormone level, and then that's followed by a trough period where there's no hormone present, and this protein binding provides a buffer to regulate the amount of free hormone in blood. And as I said, it's dynamically regulated with the rate of secretion, the rate of degradation, and the binding of the hormone to target tissue receptors. So this is shown here. So you can see the rate of secretion and the rate of degradation are two factors regulating the amount of free hormone, as are the amount of free hormone binding to its receptor. And you can see too that hormone binding to its binding protein is reversible. This can be, the rate of dissociation will increase at times or when hormone secretion is being reduced. When hormone secretion is high, the rate of hormone binding to the binding protein will increase. So in this way, the concentration of free hormone is buffered. So if we look at hormone secretion, it's dynamically regulated to maintain a steady state around a set point or a range, although this range can vary during the day, or with age, or with physiological states. And this is shown here. As I said, hormone secretion is typically phasic, so all of these bars on the bottom line indicate one episode of hormone release. And you can see that following hormone release, the concentration in blood rises, but is then followed by a decay, a trough period, where the hormone level may be very low, may not even be detected. You can also see that if you increase the frequency of hormone release, you can change the mean level in the bloodstream. And this is one way in which the set point changes throughout the day. Now, the way that secretion occurs is usually in response to feedback, and the feedback usually comes from the action of the hormone on target sites. And two sorts of feedback, negative feedback and positive feedback. Negative is the usual system which will inhibit hormone secretion when circulating levels are high, but it will increase hormone secretion when circulating levels are low. Positive feedback is much less common, but it does allow discrete events to be rapidly reached, following which a catastrophic collapse of the system occurs, or then you have the intervention of an inhibitory pathway to terminate that pathway, positive feedback pathway. So this is shown schematically here. This is a negative feedback system. Simply, an endocrine gland will cause a hormone to be released into the blood. The hormone will act at its target site, producing some biological response. Often it's a product which is released into the blood, and when the concentration of this product exceeds a certain threshold, it will turn off the endocrine gland. But by the same token, when the product that's being produced falls below a certain threshold, this will reactivate the endocrine gland to produce more hormone. So negative feedback is not just negative, it's energizing when the level is low of the feedback signal. This will actually activate hormone secretion. Positive feedback, very simply, the endocrine gland produces a hormone, acts on its target tissue to produce a product, which is often another hormone, and this will activate the endocrine system to produce more hormone, to activate the target tissue, etc. So this is a positive feedback system, and I'll be mentioning two examples of positive feedback in the next lecture. Feedback itself, as I said, reflects the action of a hormone on target sites. And the first requisite for hormonal action is the binding of hormone to a specific tissue receptor. And all cells in the body are exposed to all hormones in blood, but it's only those cells that have receptors that will respond to particular hormones. And this is shown here. In blood, you find many, many signaling molecules, and of course all cells are exposed to blood, but it's only those cells that have receptors for a particular molecule that will respond to that molecule. Now, if we look at receptors, the best way to envisage a receptor is actually as a lock, which is being opened by a key, which is the signaling molecule itself. A hormone is the key for a hormone, a lock key scenario. Now, hormone receptors have a number of types. There are membrane bound receptors that face outwards into extracellular space, and these typically are the receptors for protein hormones. And because protein hormones cannot cross the lipid bilayer around the cell, they need second messenger systems which transduce the message of the hormone to induce biological responses. And these characteristically are rapid, short-lived actions that are mediated through the activation or the inhibition of pre-existing enzymes, and typically these result in the amplification of intracellular cascades. And a typical example is this one, where the second messenger is cyclic AMP, which is produced as a result of the action of this enzyme adenylate cyclase, which is membrane bound. So you can see that activation of this receptor, this membrane receptor, activates cyclic adenylate cyclase, results in the accumulation of cyclic AMP, which will then go on to activate protein kinase A, which will phosphorylate proteins and result in alterations in cell function. Now, I know that you all know cyclic AMP is the second messenger. It's part of Biology 30. What other second messengers do you know? There are many second messengers. Diacylglycerol, inositol phosphate, yeah, triphosphate there, that's another two others. What else? Calcium, yes, that's the second messenger. GMP, yes, cyclic GMP. Yes, so you're aware that there are many different second messengers. And you can see this is why you have a rapid effect, because of this amplification system through which one hormonal molecule results in the activation of many molecules of cyclic AMP, protein kinase A, which will phosphorylate many proteins. This is why you have the rapid effect. In addition to membrane receptors, you also have nuclear receptors. These are typically for steroid hormones and for another class of hormones which I haven't mentioned. These are thyroid hormones. Now, thyroid hormones are a unique type of hormone. They're made from an amino acid, tyrosine, but they have characteristics of steroid hormones. And like steroid hormones, thyroid hormones have nuclear receptors, and typically these actions are genomic. They involve the induction of protein synthesis. And because protein synthesis takes time to occur, these hormones take longer action than membrane-bound protein hormones. There are also cytoplasmic receptors. Sometimes they're not true receptors. They're actually binding proteins within the cell that move the steroid hormone into the nucleus. Steroid hormones are actually soluble in the cytosol of the cell, so by binding to these so-called cytoplasmic receptors, this provides a shuttle mechanism to go from cytosol into the nucleus. In some cases, these are also binding proteins, just as ink provides a reservoir of the hormone inside the cell, although in some cases, they're actually true receptors which are found on organelles like the mitochondria. As you will see, these actually mediate directly actions of thyroid hormones on mitochondrial function. So, usually thyroid hormones and steroid hormones, their receptors are nuclear receptors, and binding of the hormone to the receptor causes gene transcription, resulting in mRNA translation, resulting in proteins which cause biological actions. You can see here, it's actually the hormone receptor complex which moves into the nucleus and causes gene transcription. And here you can see that these hormones can simply cross the lipid bilayer around the cell, and they may encounter a cytoplasmic receptor, which I say provides this shuttle mechanism to move the hormone into the nucleus. Finally, I just want to say a few words about receptor regulation, because all receptors are proteins, so they're all being synthesized or degraded, and the rate of synthesis and degradation can alter the rate of receptor regulation. So downregulation is when less receptors are present, and these usually occur at high concentrations of the hormone, and the reason it occurs is to prevent overactivity in the cell. And this usually reflects decreased receptor synthesis or increased receptor degradation. You could also get less receptors if they're membrane receptors if you internalize them within the cell. And this also occurs for membrane receptors. You could also downregulate a receptor if you dissipate its signal transduction system from the receptor itself. Another approach would be desensitization of the receptor, and this occurs when the receptor has less affinity for its hormone. So the binding pocket of the receptor has a change in its conformational state. Again, this occurs at high concentration, again to prevent overactivity, and is because of this conformational change in its structure. So downregulation is shown here. When hormone concentrations are high, you find that a number of receptors that are present actually become reduced. Conversely, when the amount of hormone present is low, you find that the number of receptors present will increase. So it's a way to regulate the amount of biological activity. The other way you can get membrane receptors downregulated is this. When a hormone, protein hormone, binds to its membrane receptor, this causes the receptor to move laterally in the plasma membrane, and hormone receptor complexes accumulate in certain areas of the cell, so-called coated pits, and this causes a change in the amount of protein in the plasma membrane, which causes it to invaginate, and this invagination can become pinched off, and this was taking away the number of receptors from the plasma membrane. And because lysosomes come along and cleave the hormone receptor complexes, this is thought to be a way in which protein hormones can actually get into the cell and may have long-term responses once they get into the cell. Downregulation is one way you can alter activity. Upregulation is the other, which I just mentioned. Another way is to sensitize the receptor to increase its affinity for its hormone. And again, this will occur at low hormone concentrations to again increase activity, and is again a change in the conformational structure of the receptor. And the final thing that I want to mention is that endocrine dysfunctions can occur with problems in hormone synthesis, can occur with problems in hormone regulation, and can occur from problems in hormonal action. It could be problems with a receptor resulting in tissue resistance. So these are the three usual problems that cause endocrine dysfunction, and of these three, the second one is the most common, that you have defects in hormonal regulation. There are usually problems with feedback, which results in too little or too much hormone secretion. But I will be giving you examples of all of these types of endocrine dysfunction problems in the following lectures. Okay, so we'll start